our pleasure to have Nigel Peterson here from Emory. Nigel is a clinician and a scientist. I think it's the first one this semester. Nigel did his clinical training in Australia, University of Sydney, and then he did his postdoctoral and residency training here in the States at Harvard with the Harvard system of hospitals at Mass General, Beth Deacon's, Children's Hospital, and there's where he also learned to bridge the gap between animal studies and human studies. Nigel's an epileptologist, so he specializes in preclinical assessment for surgery for issues with epilepsy. And he's going to tell us today about the potential therapeutic benefits of monitoring and modulating large-scale neural networks in the context of epilepsy therapeutics and protocols. So, Nigel.
Okay, thank you very much, Bilal. I appreciate it. Thanks for inviting me. Thanks for being here. I really appreciate it. It's a great opportunity to be here at Georgia Tech, which I have enormous respect for. The biomedical engineering department's been a wonderful thing as well. And I've met some faculty, like Garrett, for example, and Annabelle. And also I've had the luxury of having an undergrad student who's been a rock star, and I'll talk about her work in a little while as well. So today I want to talk about epilepsy and sleep, actually a little bit. And the importance is that in neuroscience, we often study local circuits, but I think that we really need to study large-scale circuits. And that's the whole point of this talk, really, is to look at different ways of studying those, innovating to do that, and also justifying why that's necessary. So I'll start out by talking about the clinical problem, just as an example from epilepsy, and then talk about sleep-wake networks as a modulatory target for epilepsy. I'll talk about a more specific example of a particular circuit that I've been studying, and then I'll talk about the development of novel technologies for studying large-scale circuits in epilepsy and elsewhere. So the clinical problem is that 1 in 26 of us will develop epilepsy over the course of our life. It's actually a very prevalent disorder. The population prevalence is about 1% at any time. And until recently, people with epilepsy would end up institutionalized. I don't know whether anyone knows where this is in Georgia, but this is the Milledgeville Psychiatric Center. And people there were even 100 years ago, were in really degraded, this is less than 100 years ago, by the way, this is in the 1960s, and this actually from a similar institution in Massachusetts. But nonetheless, people were in really degrading circumstances in these institutions, and a lot of them had epilepsy because there wasn't treatment for epilepsy until about 100 years ago. Just out of interest, local history, 30,000 people died in Milledgeville. It's the biggest psychiatric institution in the United States. Pretty crazy. Someone supposedly accidentally knocked down these tombstones, so you can't find out how many people died anymore. But anyway, that's the history. Nonetheless, people, and because of these institutions, people really started to study epilepsy. And so Gowers is the first person that wrote in detail about epilepsy, and he was able to do this, and he was able to compile 2,000 cases of patients with epilepsy in a book, which is still worth reading and really prescient, because he was running one of these institutions, and he could just see tons of patients. They had uncontrolled seizures, they had no medications, and so he could actually observe them have seizures all the time, he could examine them while they were, and he learned a lot about epilepsy. Now that all sounds terrible, and aren't we glad that we're not living in the past? But in fact, despite the number of anticonvulsant drugs that we have increasing, it remains true that 1/3 of people are medically refractory to medications. So 1/3 of people continue to have daily seizures or frequent seizures, despite the fact that they're on medicines. And those 2/3 that are treated with medicines often have side effects. And so, yes, we've made a lot of progress in the last 100 years, but it's not great. So what happens to that 1/3 of patients that aren't treated at all with medicines is they go through a surgical pathway eventually. But this is the general lay of the land with regards to what happens. You start one drug and 47% of people respond, often people with generalized epilepsies, which are where they have an unexpected seizure without a warning and they have a convulsion. And then when you get to the 3rd or 4th agents or others, you still have a lot of people having uncontrolled seizures, 1/3, and most of those are people with focal epilepsy. And so that means the seizures, at least putatively, start in one part of their brain and then spread, and that also makes them a candidate for surgery. Just thinking about the social impact of epilepsy today, SUDET, which is sudden unexpected death in epilepsy, which is where people have a seizure and die unexpectedly because they're face down or they can't breathe, or sometimes just because probably their brainstem depolarizes and autonomic function's impaired. It's actually the 2nd leading cause of productive life years lost after stroke for neurologic disease. Because a lot of these other diseases happen later in life. Stroke, of course, is just very prevalent, causes a lot of disability. This often happens to people in their 20s and 30s, and they die, so there's obviously a huge loss of productive life years. Now, just I want to forecast to preempt the discussion about sleep-wake by saying that most of the people that die from sudden unexpected death in epilepsy in hospitals, because it happens there, it's very hard to resuscitate some of these people, that it happens during the night or during sleep. Okay, so there's a really thought to be a strong association with maybe REM sleep or maybe non-REM sleep, and it really doesn't happen much during the day when people are awake. And so we'll come back to that. But I just want to talk about the surgical treatment again now. So uncontrolled epilepsy can be treated surgically, and there's a lot of different surgical options. I won't go through these in great detail, but basically it amounts to either removing tissue or destroying tissue. There are more invasive and less invasive ways of doing that. Emory, which I'll talk about in a second, is a center for minimally invasive investigations in neurosurgery and epilepsy, so it's something that we're proud of. And neuromodulation is a possibility as well, so stimulating to prevent seizures or to abort seizures is also a possibility. The treatment of generalized epilepsy, which is about a quarter of those patients that don't have well-treated seizures, is still a problem, and it's still essentially experimental. So we have something to be proud of here, and including a couple of the labs here are part of Georgia Tech as well. So Garrett Stanley's lab's mentioned here as well, but we have a very large number of basic science labs in Atlanta studying epilepsy. It's probably the largest concentration of epilepsy basic science labs, at least that I know of. I think some places like UVA comes close, maybe Stanford has a few labs, but actually we numerically have a lot. So more than 12 labs studying epilepsy in some way or another. And we have a highly active epilepsy program as well, clinically, which for people like me presents a problem because there's so much clinical work that sometimes it's a bit of a distraction from being academic and doing research, actually. But we do about 45 implantations per year intracranially, which is the kind of basically enables you to do electrophysiology in people, right? And I'll talk about some of that work later. And we have about 1,000 new visits a year, and et cetera, et cetera. So it's a pretty big center. Now, despite all the technologies that are available for neuromodulation and surgery, the outcomes are still terrible. And this isn't just our center, it's actually universal. That if you start to look at patients who have extratemporal lobe, meaning seizures starting outside their temporal lobe, and they don't have a lesion, then the outcomes are horrible. It's like about a third of them have effective treatment of seizures with surgery. So imagine going through all the process of trying all these medicines, you're still having seizures, you go through the process of intracranial monitoring and then brain resection, and you still have seizures, right? There's a 2/3 chance that you'll still have seizures in this circumstance. So that really encourages us to ask why that is. Like, what are we missing? What's the problem in epilepsy that we don't understand that's causing these patients to continue to have seizures? So probably some of the common ones are taking aside possible genetic contributions, which are there. I can talk about that if you're interested afterwards. But really, these are the kinds of things. So we don't take enough tissue. You know, there's evidence that if you take more brain tissue, that the patient does better, but then of course they may have worse outcomes from a cognitive standpoint or an emotional standpoint. The epileptic network might be wider than we appreciate, and that's something I'm going to show you is definitely the case, and that's something we need to really work on. Large-scale network hyperexcitability can be translobar. So sometimes you'll have a patient who has a frontotemporal epilepsy, and you just really don't want to take their temporal lobe and part of their frontal lobe at the same time. Some centers do, but it's pretty aggressive. And then finally, and this has been, I think, a big problem in North America, is not really actually implanting the brain in a way that we can study the seizure network. So if you put the electrodes in the wrong place, you're going to think the seizure starts in the place where it first invades the electrode network, not where it actually starts in the brain, right? So that's a huge problem. So I'm going to just talk about history briefly and why that might be the case. And I think probably most of you would know about, or maybe I'll skip here, the homunculus, right? So everybody in Neuroscience 101 hears about the homunculus. And it was described by Penfield and Jasper specifically, I guess more Penfield. And actually, if you look in the German literature, it was described by his mentor, Ottfried Förster. So I don't think Jasper and Penfield were quite as original as we think they are in the English literature. But nonetheless, this is the kind of thing you can see. Now, this was meant as a cartoon to show you that there is a topographic or somatotopic representation of the body on the brain's surface, but it doesn't turn out to be quite like this. It turns out to be multiply overlapping. There are multiple areas that represent each area, et cetera. It's much more complicated. And it was described in this book, which is really great. If you can get a hold of it, It's a great book to read. But maybe the history is interesting. So Penfield, who's seated here, he wrote some really interesting books, including a fictional treatment of the life of Hippocrates, which is really interesting, called The Torch, if you want to read some of his stuff. He was a pretty interesting guy. He studied with Ottfried Foster, and he brought the technique of doing these kinds of craniotomies and grid implantations and 50 hertz stimulation back to North America. He worked with Herbert Jasper. Now, Herbert Jasper was a brilliant neurophysiologist, PhD, wasn't a neurologist, but he would be in the OR with Jasper, with Penfield, recording the patients and doing stimulation studies in the patients. Now, that resulted in a practice essentially like this. So doing really large craniotomies to put in grids. Now, what would be the problem with that? So the problems are, the obvious problem is, do you really want a craniotomy like this? No, no one does, hopefully, right? The second problem is it can be morbid, right? So some patients develop dural bleeding or edema of the brain and actually can be fatal, and it's pretty uncomfortable. And then the last problem is that most of the brain's in the sulcus, right? So it's only a third of the brain's surface that's actually on the crest of the gyrus, and 2/3 are in the sulcus. And then you can't even access the basal and medial aspects of the brain very easily. So basically, you're really undersampling in a vast way, and they didn't know it at the time, but now we know that many epilepsies start in the sulcus because that's where the cortical malformations are. So this is a problematic technique. Now, in parallel, and interestingly, a very different team of people developed a completely different technique. So in the 1950s, Jean Tellerak and Jean Banco developed a method for putting depth electrodes in the brain instead. Now, understanding their background is important. So it turns out that despite John Tellerak being a neurosurgeon and being very famous for stereotaxic surgery, he was never trained as a neurosurgeon. He was a psychiatrist, actually. And he was interested in psychosurgery and in physiology. And so he trained himself to put in depth electrodes in patients. And it's pretty cool because they had to work out a way to image the brain before MRI or CT were invented. And so at the St. Anne Hospital in Paris, they cleared the chapel, and because it was the longest room in the hospital, and they put the patient at one end of the room and the X-ray source at the other end of the room so that they wouldn't get a lot of parallax error when they imaged the patient's skull and did fluoroscopy with the patient. So it's pretty cool. Like, I don't know whether you can imagine that happening here, that a psychiatrist is suddenly doing neurosurgery and we're doing it in the chapel, we're using X-ray, right, and making X-ray films. It's pretty cool. But in any case, Banco was also a very classically trained neurologist, and he was trained in the tradition of Charcot, which is very much about anatomy and localization. And so their approach was very different. And so they said, seizures spread through brain circuits. They are localizable phenomena, right? So if someone has a seizure, we can work out, based on localization theory, where in the brain it's coming from. And so Banco had this approach, and Tellerak was prepared to put electrodes wherever they could safely put them. And it resulted in them having this clinico-electro-anatomic approach. That's very different than the North American approach to epilepsy investigations. So I'll talk about that a bit more in a moment, but these are the people that really, well, particularly Patrick Chauvel, he's an important influence on me. I see him frequently, and he is one of Banco's students, and he's really one of the foremost people in doing this kind of correlation of localization with the electrical phenomena and anatomy of the brain. And he brought this technique to North America when he was forced to retire from France, right? You have to retire when you're 65, and so he thought, I'm not going to retire. So he's been working in other countries. He's working in Australia now as well. And this is the, our neurosurgeons, John Willie and Bob Gross, now use a robot as well. So robot-assisted stereotaxis to put in depth electrodes, which that makes things faster. It doesn't really make things more accurate. And it looks like this. And so instead of having a giant craniotomy, it's possible to actually shave the scalp, at least in the area of putting depth electrodes, drill small holes and put depth electrodes into the brain. And it's really much better tolerated by patients. So after 2 or 3 days, the headache goes away. They have a bit of tethering of their jaw muscles with these temporal electrodes. But generally speaking, they're pretty comfortable. And they can stay in the hospital for a long time while we record them. And most importantly of all, it actually samples the right part of the brain, right? It's not missing the sulci and the medial and basal cortex. So say, for example, you had a patient who, when they had seizures, they experienced an intense sense of fear. And they had all these autonomic signs, like they might have piloerection or pupillary dilation. And this is how the seizure unfolds. And it's very important to understand the temporal sequence and the timing between these different semiologic features, as we call them, to localize in the brain where the seizure is coming from. And then when you record this patient intracranially, No surprise, they say they're feeling scared and they have autonomic signs. So generally speaking, it's not uncommon that this would come from the amygdala. And so here you can see the A electrode is amygdala, and the medial part of it's here. And you can see the beginning of the low voltage fast activity as their seizure evolves here. And then you can spread the pattern of spread through their brain as well. So it's a pretty cool technique. But what it enables us to see, and I think all of the people that use this technique would agree, that seizures aren't really just a local phenomenon. And so you can see in this patient of ours that this is a patient whose seizure starts in the hippocampus. So this is sort of interictal discharges here. And the seizure is starting somewhere around here, where you have a herald spike, and then you have this low voltage, fast evolving spikes, and then it continues with this higher frequency activity here, and it propagates through these networks. And so here it's propagating into the basal temporal lobe and the temporal pole, for example. Okay, so it's nice physiology. But the interesting thing here is that if you then stimulate parts of this network, multiple components of this network are hyperexcitable. And so in epilepsy basic science, for a long time, people have studied the trisynaptic circuit of the hippocampus itself, but it turns out in our patients, we really are worried about the basal temporal lobe in this patient, or the temporal pole, or larger networks, and sometimes even in the orbital area, for example. And so this means that the hyperexcitability and the epileptic abnormality is much wider than that local circuit that we typically study in our mice. And it presents a problem for surgery too. How much of this hyperexcitable network do we need to remove? Well, it turns out the more the better, but then we have to put limits on that as well from a judgment standpoint. Often we can do stage surgery these days, which I can talk about if you're interested. And so really we need to study more than the network. I guess I've already made these points. So what I want to talk about now is I want to talk about an example of a kind of thing that we could think about. So if we think about large-scale networks, the really absolutely perfect magisterial example of this is the sleep-wake network. So it controls the whole brain, really, right? Brainstem activity and cortical activity, everything else changes with state changes between sleep and wake. It also turns out, interestingly, of course, that the sleep-wake network changes brain rhythms. And epilepsy is actually an abnormality of brain rhythms. It's about synchronization and hyperexcitability, and there's nothing that affects that more than these modulatory networks. So I'm going to talk about a little bit about my history and evolution during my postdoctoral fellowships doing sleep-wake neurobiology and I hope to bring that to bear in epilepsy. So sleep-wake was of interest in the earliest days of epilepsy, but it really hasn't been studied very much. And this is from the book that I showed earlier from 1880, 1900, the different editions, showing that these patients in aggregate would have very characteristic times of day or sleep-wake state that they would have their seizure in. So it's very common for patients to say, I have seizures when I wake up a few minutes afterward, right? Or they have seizures only in sleep. And oftentimes you can even find out which part of the night it's in. So it might be when they're having slow wave sleep would be a common thing for frontal lobe seizures. And there's lots of data to suggest this. In this case, this is just showing abnormalities in different brain regions in a patient with and without epilepsy in different stages of sleep. But oftentimes in a patient with a hippocampal epilepsy, we can see a sleep facilitation of discharges. And so suddenly the record is absolutely filled with discharges as the hippocampus has made a state change. There's also evidence from the NeuroPace device. So NeuroPace is a small device that engages in closed loop stimulation. So it senses and it can stimulate the brain when it detects a seizure or discharges. But it also has a recording function, so it can record episodes of discharges, and then the patient can use a wand to upload that data to a database and we can review it. And hopefully they upload it every day, and if they do, we can make some useful conclusions about it. But this is just showing different kinds of epilepsy here. So this is medial temporal, neocortical, and frontal lobe epilepsy, and then showing the different distribution of seizures over the circadian period. So it's just telling you clock time because, of course, you don't know whether the patient's awake or asleep when it comes to this device. We've already spoken about why this matters because of SUDEP, right? So there's another line of evidence. And generally speaking, I think that this is a sort of philosophical point, but people often describe the propensity for someone to have seizures in terms of their electrophysiological background. But you could argue that what they're saying is the EEG state determines whether they have seizures. But you could also argue that that's an epiphenomenon, really. It's not really a very satisfying explanation to say that, yeah, they have rhythmic slowing or something at this stage. But really understanding what that means in terms of local circuits is actually important as well. So my background was participation in research about the arousal system principally, and also about REM sleep. And so these are some of the papers from the lab that I was in as a postdoc. And what we were interested in was uncovering, identifying specific cell types that participated in arousal. And so in my case, I was interested in hypothalamus and basal forebrain, and I can show you some of that work in a little while. There are very, very few studies that show the contribution of sleep-wake systems. So despite the fact that the sleep-wake network sets brain rhythms and cortical excitability, it's almost completely unstudied in relation to epilepsy, surprisingly. So this represents a great opportunity. These are some of the results summarized from what we know, but there's very little. So does that make sense in the sense that if we were to manipulate the sleep-wake system to treat seizures, well, that may sound terrible. I mean, maybe it would stop people from ever sleeping and therefore they won't have seizures, or make them sleep all the time so they don't have seizures. That's not really very helpful. But it turns out that there is a lot of nuance in the sleep-wake system, and so it's possible to manipulate components of it that set excitability or rhythms without causing disruption, as far as we know, at least to mice. And this is still an active area of research. So I'll give a specific example here. And so frontal lobe epilepsies seem to occur in the context of a lot of frontal lobe slowing, so delta activity in the frontal lobe, rhythmic slow activity of neuronal firing. And so it turns out that if you, this is from a larger study where we're using dreads. So who here has heard of dreads? Chemogenetics? Yep. And so in this case, we can introduce a designer receptor into a neuron that is affected by a designer drug but leaves everything else intact, or is inert in other cell types. And we can do that in a cell-selective way. So here in a cell-selective way, we've put an excitatory designer receptor into GLUT neurons, GABA neurons, or cholinergic neurons in the basal forebrain of the mouse, and then studied the effects of activating those different neuronal populations. So it turns out it was a mystery. You know, people thought that cholinergic neurons promoted wakefulness, but actually it turned out that the most wake-promoting neurons were GABA neurons, and so I'll show you that here. So here this is a mouse where those GABA neurons have been transduced with that designer receptor, and here we're just giving a saline injection, and here are Fourier transforms. So this is 0 to 30 hertz, and this is a time base here of 12 hours. Here is 150 to 0 hertz here as well. And then you can see the neck muscle myogram here as well, which gives you an idea about the behavior of the mouse. And then this is what's called a hypnogram, so it shows you the sleep-wake state of the mouse. So when it's down here, it's wake, then it's slow-wave sleep, and then there's a little bit of REM sleep here. So this is, unfortunately for a mouse, this is what normal sleep looks like in a mouse. It's very fragmented, at least in an impoverished environment. But when we give CNO to activate that receptor, we get sustained wakefulness. So in this case, the mouse stayed awake for about 6 hours before it went back to its normal sleep-wake cycle. So this suggests that actually it's the GABAergic neurons in the basal forebrain that are important for wake, and they probably do this by disinhibiting cortical circuits by their projection to Purkinje neurons in the cortex. The other population that was classically thought to be more involved in wake were cholinergic neurons. And if we, in a like manner, if we activate them with clozapine and oxide, then it turns out that the sleep-wake behavior of the mouse doesn't change at all, but you can see that there's an almost total loss of delta activity in the cortex. So just to show that a little bit more clearly, here you have the slow wave activity, large amplitude slow activity in the cortex in a mouse that's sleeping. And then when we give clozapine and oxide, that actually goes away despite seeing the same EMG and behavioral changes associated with sleep. So in this case, the total amount of sleep hasn't changed. All we've done is get rid of that slow activity in the cortex. Could this be anti-epileptic? It's possible. And that's something that we're in the process of studying, actually. So now I'm going to talk about a specific circuit. So I'll talk about medial temporal lobe epilepsy. So medial temporal lobe epilepsy is actually involving the hippocampus and its related circuits. It's the most common kind of focal epilepsy in adults. It's about a third of patients overall, and it's the most common medically refractory epilepsy. And it turns out that there's some good treatments, and so Bob Gross was instrumental in bringing laser ablation to Emory, so to laser ablate the hippocampus and the amygdala and part of the uncus and entorhinal cortex to treat these patients, and that's actually pretty effective. So probably about half of the patients are treated well by that. But then there are other patients that aren't, and we think that's because a larger network's involved. So having other approaches to this is important. So I'll just show you some examples of what this can look like. So the seizures with medial temporal lobe epilepsy can be quite varied. There is a sort of a syndrome of semiologic features that we see.
Oh, help me, help me, help me, help me. Oh, me. Please help me, please.
And you see he's making this funny chewing movement with his mouth. Sometimes there's some utilization behavior, so if you put something in front of them, they'll futz with it with their hand. In this case, probably this is utilization behavior, but it's off camera.
Touch your nose.
So he can do it. So his seizure's probably ended. But he wouldn't have lost awareness, and his language was probably intact, although there wasn't a good ictal exam. So it's very important to examine a patient when they're having a seizure so we know what's working and what isn't. It's very, very hard to do. It means someone has to rush into the room immediately and start doing a neurological examination on them, and it's actually pretty hard to organize. In any case, this is one example. I'll show you another example. So this is another patient with a similar brain localization of their seizures, but a different looking syndrome. So here she probably knows something's happening, so she's pressing the button there. She doesn't say anything, and now she's just staring. So it's another whole talk, but if you're interested, the idea of studying consciousness is really interesting in these patients because at some point they're obviously offline. They have a behavioral arrest, they're amnestic, but they may also not be conscious. So now it's spreading into her motor cortex, and you can see that her glottis is closing, and her mouth is twitching, and her head's elevating, and then she goes on to have a convulsion, actually. So the manifestations can be varied, but these are the kinds of things that we're talking about. This is very debilitating for people. I mean, can you imagine this happening when you're at work? Turns out a lot of patients with epilepsy end up losing their jobs, even though that's, of course, not correct labor force behavior, but that's very common. So what happens in these patients? So if you're lucky enough to get to remove their hippocampus, and you look at it down the microscope, then you'll see these kinds of cellular changes. So this is a normal hippocampus, no hippocampal sclerosis. And this is the most common type of hippocampal sclerosis. And you just see that there's vast cell loss. And it turns out that this dentate gyrus cell loss is also associated with their cognitive and memory difficulties as well. And so not only the seizures are a problem, but these patients can often accumulate some pretty significant cognitive problems related to cell loss in the hippocampus in medial temporal lobe epilepsy. And so these are the canonical hippocampal circuits, and I'm sure that Annabelle could speak at great length about much more detailed elaborations than this, but this is a very sort of simple overview, and this is often what one learns about at it's Neuroscience 101, is this is the classical trisynaptic circuit. And there are a variety of different circuits, but it turns out that the key structure here is probably the dentate gyrus, at least when we're thinking about this local circuit. So the dentate gyrus, I'll show you the picture again here. So the dentate gyrus is sitting here, and it's actually the input region for the hippocampus proper from the entorhinal cortex. And the model is that these cells are very hyperpolarized. They don't fire very easily, and they probably act as a kind of gate and a filter for activity getting into this otherwise amplifying circuit of the hippocampus. So it's very important to have this dentate gate intact, we think, in epilepsy. And it turns out that the inhibitory neurons in the dentate gyrus that are responsible for part of this hyperpolarization of dentate neurons, granule cells, are lost in epilepsy. And so you can see there's cell loss here to here. And this is in a rat model of epilepsy. And there are also excitatory recurrent synapses that form onto the dentate granule cell. And so they actually hyperexcite each other. They wind each other up. And it turns out that if you inhibit so this is using optogenetics, and this is from Esther Cook-Magnuson's work. But if you inhibit the dentate gyrus during a seizure, you can actually abort the seizure. So that's an example of a seizure in a mouse that has a canic acid model of epilepsy. So if you inject canic acid, it's hyperexcitable, they go into status epilepticus, so they develop seizures. They have spontaneous seizures. If you wait for a spontaneous seizure and then turn on the orange light, then you can actually terminate the seizure. If you, in contrast, wait until there are some discharges and you turn on an excitatory opsin, in this case channel rhodopsin, then you can make a much higher grade of seizure as well. So you can facilitate the seizure just by activating the dentate gyrus. And I think most interestingly of all, if you have a mouse that doesn't have epilepsy at all, if you excite the dentate gyrus, you can actually cause what look like spontaneous seizures. So we think that the dentate gyrus is very important in epilepsy. Some people even think that it's the seizure onset zone in humans and some animal models, at least in certain cases. Now, the reason that I'm talking about this is because it turns out that a node of the sleep-wake network modulates this, or a couple nodes, depending on the way you look at things. And it has very, very few inputs. So the dentate gyrus actually does not have very many inputs. It has input from the entorhinal cortex, we know. It has input from the septal area, which is indirect. So the septal GABAergic neurons project to GABAergic neurons in the dentate gyrus. And then it has a direct input from the supramammillary area as well, of the hypothalamus. And this is relatively unstudied. And the interesting thing here is that these neurons, maybe inhibitory, maybe not, I'll talk about that in a minute, directly innervate the dentate granule cell and the proximal dendrites of them. So they may actually have a role in filtering information coming into dentate granule cells. And it turns out that these terminals that arise in the hypothalamus undergo clearing and then dispersion in the context of epileptogenesis, meaning a rat, in this case, becoming epileptic. Okay, so there's a modification of that architecture of the synapses as well. So that's the reason for studying this area, and I'm just going to talk about a bit of my sleep-wake work in this area and then talk about how it controls the dentate gyrus in a little bit more detail. In this case, we're using dreads, and so this is a cartoon of showing when you inject the vector, then in neurons that express Cre recombinase, you can express this designer GPCR, the G-protein coupled receptor. And this is just the same thing, so we use a flex orientation vector. Here you can see in orange, you can see where the vector is being expressed. This is a very large preliminary injection, and then you can see black dots where there's CFOS activation. So you can do an immunohistochemical way of looking at neuronal activation. If you activate neurons in the brain and then perfuse the mouse an hour and a half later, you may see FOS expression in the neurons that were activated, and it looks like this. So you see this nuclear black reaction product here. And so this is, we were studying this area because of its role, we thought its potential role for sleep-wake rather than because of its hippocampal control, but we'll get there in a second. So here, this is even more interesting than the basal forebrain, in that if you excite, in this case, glutamatergic neurons, and so here we're giving CNO, in the caudal hypothalamus, the mouse will stay awake for up to 12 hours. And so you can see, this is again a fast Fourier transform here. This is the amount of wake over time, and so it's highly significant, and it changes the power frequency spectrum of the hippocampal EEG as well. As you can see that there's a change in the theta frequency of the hippocampus. When you activate this brain structure, it becomes faster. So we wanted to know where it was exactly, and so we made smaller injections. And it seemed like the supramammillary nucleus was a good candidate. So here we get a lot of increase in wake, whereas when we activate the mammillary body, not surprisingly, we don't see much increase in arousal. And then borrowing a technique from MRI analysis, we can do non-parametric mapping analysis of the area of statistical correlation between the injection sites and the amount of wake. And it shows this is the area where the z-score is, or the false discovery rate is less than 1%. And it's an area that involves the supramammillary nucleus, and potentially part of the VTA it may be. We're trying to resolve that. Now, one of the, as an aside, it turns out one of the most interesting things about this model is that these mice, even though you keep them awake for 12 hours, they never have a sleep rebound, either by slow wave power or by sleep time. And so that's really important. It's a digression, but it's important because we don't understand the mechanism of sleep homeostasis. And so this is a potentially interesting model for that. Other manipulations that keep mice awake do result in rebound in both sleep time and slow wave power. And so here you can see the accumulation of sleep time. So this mouse is awake and it never catches up. The lines don't come back together. This is looking at the rate of accumulation. And then this is looking at this, hang on over here, sorry, is looking at the slow wave power. So it's important because mice rebound not just by the amount of sleep, but also by its intensity, by its amount of slow wave power. And there's not really a difference between these groups as well. This model's been replicated recently. I won't go into this, but it turns out it's an interesting model too. So one question is Alzheimer's disease affected by sleep deprivation? And in this case, this is about tau accumulation in an Alzheimer's model of a mouse. And it turns out that if you keep them awake for large amounts of time, then they accumulate more tau protein. And this was from Dave Holtzman's group at WashU. So what happens if you turn these neurons off? Because after all, if you activated the dorsal horn, the sensory neurons, you'd probably keep an animal awake, right? Because it would be itchy or it would feel something. So it means nothing to turn something on. You have to turn it off and show that there's a decrease in wakefulness as well. And so that's what we do here. And it turns out the effects actually looks modest, but in terms of these distributed nodes of the arousal system, this is about as good as it gets. So because there's a redundancy probably in the arousal system. I might skip over some of this actually and get to the most interesting part. So there's another population of nitric oxide neurons that are probably involved in REM sleep. But let's go to this. And so we were wondering, there's a population of GABAergic neurons in this nucleus. And so we thought that if we turn on the glutamate neurons, we would get wake, and if we turn on GABA neurons, classical local interneuron thinking, we would get an increase in sleep, right? Well, it didn't turn out to be the case, and so we did a double staining for VGAT, which is a marker of inhibitory neurons, and VGLUT2, which is a marker of, this is showing NOS actually, and VGLUT2, which is a marker of excitatory neurons or glutamate neurons. And so the cyan color here, or the magenta color rather, is the excitatory neurons, and the green color are the GABAergic neurons. And it turns out there's a population of neurons that release, well, they have markers for both neurotransmitters, both GABA and glutamate. That's pretty unusual, right? Like, do you know of anywhere else in the nervous system? Yeah, well, it turns out they're actually, this population's a little bit larger, extends to another area, but it's the only area in the brain. So this is, what I've done here is I've just, wherever there's an overlap of the magenta and the green, I've enhanced it to white so you can see this more easily, but they're both there. But the question, of course, is does that really matter? Do they really release both neurotransmitters at the same time? And why do we care when we're talking about epilepsy? So it turns out, as I'll come back to, that these neurons plaster the proximal dendrites of dentate granule cells. And so these are the very neurons that project from the hypothalamus to the dentate gyrus that I was talking about before. There aren't many inputs to the dentate gyrus, and this is the one, and it contains both neurotransmitters. Isn't that crazy? And so the experiment here to show that both neurotransmitters are actually released is to do what's called CRACM, or channelrhodopsin-assisted circuit mapping. And so we can inject channelrhodopsin into the supramammillary nucleus, and it will express in the neurons and the terminals as well. And then we can flash a light while we're recording from the postsynaptic neuron to see what's released. And so this is what we see. So we see that in the presence of bicuculline, you get this fast, actually turns out to be AMPA-mediated synaptic. And in the presence of an AMPA block, you get a similar postsynaptic event. In the presence of both, you actually abolish the postsynaptic potential completely. And then I'll just skip forward a little bit. So this is just showing that the morphology, the timing, and the reversal potentials are consistent with these. But the most important thing is that the onset delay in milliseconds to the postsynaptic neuron is really short. And so for electrical stimulation, say for example you were electrically stimulating the perforant pathway and recording from dentate granule cells, then you would expect a 2 to 4 millisecond postsynaptic delay. In the case of channelrhodopsin, actually, because it actually has to get calcium into the cell and then release transmitter, it's a little bit of a different mechanism. This is an accepted monosynaptic delay. So it doesn't matter whether these neurons, whether you express it in GABAergic neurons or glutamate neurons in this region, you get this postsynaptic potential that contains both GABA and glutamate. So indeed, these neurons are capable of releasing both neurotransmitters. Another group has shown, interestingly more recently, that they're in different pools of vesicles. And so one can imagine they may have different release probabilities as well, and that actually may be susceptible to modification. So it's very interesting, something to explore. And so if you look at the projections of these neurons, though, they really are very targeted at the dentate gyrus. And so here's the neuronal population. Here I've used a viral vector that's only expressed in GABAergic neurons, as it turns out. And because it turns out that all of the GABAergic neurons in that brain region also express glutamate, right? So this is a way of identifying those neurons. They project through the hypothalamus and the diagonal band, but they're heading up to the fornix, and they really just go to the dentate gyrus and CA2. So while glutamate neurons of the supramammillary area are important for arousal, and I didn't talk about NOS neurons, I can talk about that later if you're interested, and necessary for REM sleep, dreaming sleep, these GABA-glutamate containing neurons have an unclear role, but really plaster the proximal dendrites of dentate granule cells and actually target them with both neurotransmitters. So how do we explore what the role of this pathway is in epilepsy? So so I have a bunch of flox mice. So in this case, you can inject them with Cree in the area of interest, and you can delete the vesicular transporter for glutamate or GABA from these neurons. So effectively, you can delete GABA release or delete glutamate release just from this neuronal population and see what happens. And it turns out not much. And so if I just delete GABA release from these neurons, I thought they'd all become epileptic, of course. But actually, over 8 weeks of recording, we haven't seen that. So we're injecting mice to leave them longer. Previously, I'd seen in a cohort of mice that I injected, there was a mortality. And in mice, as you all know, mortality is commonly associated with spontaneous seizures as well, particularly in C57s. So we're still exploring this, but we also will explore this in the context of a perturbation. So we may need to actually give them electrical stimulation or canic acid or something, and then they may become more easily epileptic. And I'll come back to these key points. And so there's a bit of a problem with doing these kinds of studies in mice, and that's the controlled recording of large cohorts of mice and looking at large-scale networks. So I'm going to talk about that in just a minute, but first I'm going to pop back to talking about humans as well. Now, so in mice and humans, we need to understand large-scale networks, but unfortunately the methods to do that are not very evolved. And so we have a clinical need for this because we need to know, so say for example, you have a patient who has that fear and autonomic changes at the beginning of their seizure. It's often the case that it's not the amygdala. Well, it's often the case that it is the amygdala in that particular example, but it may be an upstream brain region that's driving the amygdala. So you need to know the amygdala inputs. So it's very important for our implantation planning to understand brain connectivity. And there are certain kinds of experiences that patients with epilepsy have that are absolutely not due to the seizure invading that local structure. So you may have heard that people have an epigastric rising sensation with a temporal lobe seizure, but that's not because their insula is invaded by a seizure, it's because their insula is being driven from somewhere else. When the insula is actually invaded by a seizure, it spreads bilaterally very quickly and generalizes. So there are a number of clues that we have that actually we need to think of what's upstream and we need to know about connectivity. Now it turns out in the case of humans that this information is really not well organized and we don't know. And so there are a number of ways of establishing this. So we can of course do neuroanatomical tracing, but not in humans, right? My mentor in Boston wants to be injected with AAV for tracing before he dies, just so that we'll finally have some human specimens. But in general, I don't think many people are signing up for that. And it's hard to know when you're going to die as well to get the timing exactly right. And so we tend to rely on primate studies. And so there's a compendium, this book here includes about 30 cases, I think 36 cases, of injections in the cortex to see where the terminal fields are, so just anterograde tracing. And there's also a database called COCOMAC, which is a really useful resource because it will tell you which papers to look in if you're interested in a particular cortical structure to look at the tracing studies. So a sophisticated SEEG person, stereoelectroencephalographer, doing these kinds of pre-surgical implantations and planning them, looks at this kind of data to try and work out how to do the implantation. But generally speaking, it's really difficult to do that for every patient and to go through dozens of papers about primate tracing. And so there are other ways of doing this, right? And there are other ways of thinking about connectivity. And so we're talking right now about structural connectivity with neuroanatomical tracing, but there's also functional connectivity. So say, for example, you do an fMRI study and you can see that slow, bold fluctuation in the paramagnetic signal of oxygenated blood over time, and you can correlate that between brain regions. Now, the nice thing about that is you're sampling the whole brain. The bad thing about that is the time course is horrible, and you don't know that they're directly connected structurally. And so it may not be useful for epilepsy planning. Now, the third kind of connectivity, according to Carl Friston, who's reviewed this, is effective connectivity. And effective connectivity is where you can perturb something and see the effect in a different brain structure. So an example might be Annabelle's mentor in Boston invented this technique for temporarily interfering electric fields. And so you might be able to stimulate a deep brain structure with a non-invasive stimulation, which is pretty cool, actually. And then look at its readout in other electrodes in the brain. So you could look at effective connectivity that way. And of course, we have diffusion tensor imaging to supposedly look at structural connectivity. Now, there are some problems with diffusion tensor imaging. So we don't know the directionality of the connection. If the axons leave at 90 degrees, which we know that they like to do, then we can't follow them. It's difficult to follow into gray matter. And if axons are coming and going from the same fiber bundle, we can't follow them. And so generally speaking, we haven't discovered much. It's been useful for graph theoretic analyses of connectivity. That's very important because probably we should be thinking about the brain as these neighborhoods within a larger network rather than thinking of, not thinking that way. But it doesn't really tell us what the precise structural connectivity or directionality is. This is, of course, an example of fMRI here as well. And so another method that we are excited by is looking at cortico-cortical evoked potential. So in this case, we have the electrodes implanted in the patient's brain. We can stimulate one contact in their electrode array and then record the effects throughout the rest of the contacts. Now, the problem with this, unlike fMRI, is that we can't look at the whole brain, right? We have a sparse sampling problem. But nonetheless, we can see what's connected effectively. So this is a form of effective connectivity analysis. And this was first done by an absolute legend, Mary Brazier, who I think is not adequately lauded for all the cool things that she did, in 1965. So these were handmade depth electrodes. They put them in the amygdala and hippocampus of a patient at UCLA, and they stimulated the amygdala, and they saw what happened in the hippocampus. So this is really the first time it was done in this way. And then really it wasn't until the early 2000s that people resurrected this technique I should mention that in the French literature around this time, a little bit later than that, people were doing a similar thing, but it wasn't really, it appeared in an atlas and it wasn't really super well organized. And this is really when people started to study this with interest again in North America. And this is Riki Matsumoto, who's now in Kyoto, and Dilip Nair, who's still at Cleveland Clinic. These are collaborators and friends of ours. These really are critical given the limitations of DTI. So if you want a nice article criticizing DTI, comparing it in a primate to neuroanatomical tracing, then this is the paper to look at by Doug Rosine. And it turns out to look at this connectivity, we do low frequency stimulation. So here's an example of one of my patients where we're stimulating with 1 Hz stimulation. And you need to do that because the evoked potential unfolds over about 500 milliseconds. And so you need to be at least that far apart with your stimulus pulses. In contrast, good old Wilder Penfield's 50 Hz stimulation is often done for mapping. Now, funnily enough, why does he use 50 Hz stimulation? So it turns out that's the line frequency of his mentor in Germany, Ottfried Förster. And he just sort of slavishly copied that. And he came to North America, and he actually had to create a device to create 50 Hz stimulation, right? Rather than just using line frequency, which probably would have been fine. Probably would have been easier just to do 60 Hz stimulation, right? But when you do this, in any case, there are a lot of after discharges, and you can cause seizures very easily. So you can use this for motor mapping, so we can see what twitches and moves and where patients feel things. And it turns out the receptive fields are smaller, so this is much nicer for mapping. These receptive fields are big because you end up recruiting hyperexcitable networks in the case of this patient. And it's very safe. And so all of these colored boxes here where you see after discharge or runs of discharges, which are essentially potentially going to cause a seizure. Here they're actually seizures. This caused none throughout the whole thing, right? So it's very safe. We like to do it. We can map the patient's sensory motor cortex. And we also get this information about connectivity. And so my postdoc, Adam, he's a second-year epilepsy fellow and postdoc and instructor. He has this weird status like people do. He's fantastic. And so he's one of the authors of MATLAB for neuroscientists, and so I'm really fortunate that he's in my lab because I'm not that great with MATLAB. In this case, the question is, can we use this information to try and improve our planning of resections in patients? And so in this case, there's a patient, and you can see here there's subtle heterotopia here. So they have 2 abnormalities of their cortex, and these are commonly associated with epilepsy. And these are the numbers of electrodes, and so 9 and 35 are sampling the posterior heterotopia and 7 the anterior one. You can see they're projected onto the lateral surface here. When a seizure starts, it really involves electrodes 29 and 31 and 39. And when we look at the connectivity matrix, we actually found that the anterior heterotopian actually connects with the nodes in the seizure network quite well, whereas the posterior one did not, and other nodes that we had recorded from didn't. So the idea is that, at least in theory, the network's connected in such a way that it could drive the seizure from those heterotopia into the cortex and cause the seizure onset. That doesn't look like it's the case here, but then if you do a wavelet transform here and you look at there's actually really high frequency activity right at the beginning of the seizure in the heterotopion. Now you can imagine the neurons in that heterotopion are really disorganized. So you don't get these nice giant field potentials like you do in nicely organized cortex where all of the dendrites and neurons and axons are aligned with each other. And so it turned out in this patient, when we just did a radio frequency ablation of these 2 heterotopia, they've been seizure free for 18 months now. And so despite looking at the seizure and not really being entirely convinced, when we did the network analysis and then made an ablation, we were actually able to cure the patient of their epilepsy. And so this approach you can take to multiple patients, and then in case we've just submitted a paper about 10 patients. And so here, if you trigger these evoked potentials from inside the clinically determined ablation zone, so what the surgeon's planning to target for surgery based on clinical grounds, that there's a greater number of times when that's a statistically significant correlation to the seizure network, whereas it's a chance correlation when you trigger them from outside the ablation zone. And when looking at all of the patients, the yellow area here is the area that was ablated with, in most cases, with laser ablation or with radiofrequency ablation, and in no cases resection. Then that often correlates with the red stars, which are the area of predicted, suggested resection based on network connectivity. So it's not perfect, but overall it's statistically significant, and it may be an extra piece of information. It turns out that no piece of information in epilepsy gives you 100% accuracy. It's more a matter of combining lots of sources of information. So we need to improve this technique, and one of the things that's most obvious is using crappy clinical equipment. we don't have an amplifier that's good enough to recover fast enough to get that monosynaptic potential. And so we can do that. We can also try to do a source localization along the electrode array to work out what the sources are, so that then we have fewer sources to compare rather than comparing contacts. So this is just looking at the evoked potential in using different montages. So in this case, you can see that you can localize the evoked potential quite well using Laplacian sum. So this is where you're comparing the contact to its adjacent contacts, right? It's the same as doing a current source density analysis. This is using a good amplifier, and so here you can see this is 0 to 500 milliseconds, and so most components of the evoked potential are over by 500 milliseconds, as I mentioned before. These are pretty noisy traces in loaner equipment, by the way. So, but if you look at the first 25 milliseconds, you can see that there are actually significant components of the evoked potential. So this is 5 z-scores from the pre-stimulus baseline. And so there are really significant components of the evoked potential that are very early, in this case about 3 or 4 milliseconds. And so probably we can use this method of using corticocortical evoked potentials to reconstruct the monosynaptic areal connectivity of the cortex. That's the idea. So these advances will help us to understand cortical connectivity, but they may also help us to improve DTI estimates. So one of the big problems in diffusion tensor imaging and MRI is a fall off with distance. And so distance compensation metrics are really, really important. And so using this as a way to adjust parameters in diffusion tensor imaging may give us a way to improve our connectivity analyses from MRI. Because the problem with this technique is we have a very sparse sample, don't we? Whereas the DTI really wins out in a way by having the whole brain. So if we can find a way to marry these, it might be the best way forward. And probably there's also a way to improve surgical planning as well. So lastly, I want to talk about the development of large-scale network techniques for rodents as well. And so this is a slide I showed before, and this is Naomi Gudekunst, who was working on this project. And we really need a better system for chronic recording large groups of mice in epilepsy, because there's quite a bit of individual variability in spontaneous seizure frequency. And we want to place all of the headsets in one day for a cohort of mice if we can. And we want to analyze large-scale networks. We don't really want to just be recording from the hippocampus or from something like that. So the approach initially was as stage 1 of this project, which is Catherine Zhu's been working on. So she's a biomedical engineering student here. She's been absolutely amazing. So she's just starting to write her second paper now as an undergrad, which I think is always really impressive. But she took an approach which was great. So we tried a variety of different software packages to make a skull conformal head plate for mice. So the idea is that you can clean the skull, put holes in the skull, put screws in, electrodes in, and then you're kind of done. It's kind of simpler to implant rather than messing around with these things, making these things when you're implanting the animal. And so it turned out that the best software for this, because you can just put in Cartesian coordinates from an MRI of a group of mice, in the case of this was OpenSCAD. So OpenSCAD's great for people that like programming. You can just script things. So you can say cylinder here and sphere there and subtract this from that, and it has all of its Boolean logic, and you can also have loops and everything else. And so programmatically, you can create an object. And so this object is, in this case, it's about 700 lines of code, which probably some of those are not necessary for this particular version of it. And then we can use a high-resolution 3D printer, which we had to buy because the Emory doesn't have high enough resolution printers. But and this is a professional, like dental and jewelry printer. And then we can print these head plates and assemble them ex vivo. So it's nice to be able to assemble something in the lab, not at the time of surgery. You know, if you spend half an hour or an hour making a headset, that's great because it's half an hour the mouse is not anesthetized to create instrumentation. And so here, all of the components are friction fit. So we use a conductive grease to put the screws in. We just run them past the wires that go into the plug assembly. We put some conductive epoxy resin in these reservoirs and then stick the plug on, and then it's done. And we've manufactured something that has, in this case, 2 microdrives. So these are microdrivable platforms. And then also these skull screws and EMG electrodes. And the implantation is pretty simple. So it takes about half an hour to 40 minutes. So if I'm implanting and I'm working with somebody else in the lab, they can prepare the animals and just pass them to me. And it's about half an hour per mouse, which is good because that means I can implant a whole cohort of 8 or 12 mice in one day. And that's much better for variability as a standpoint. So this is just showing you the surgery. So you just clear the skull, drill the holes in, lower the headset, drop the screws in, wind the micro wires down into the hippocampus, tuck these EMG electrodes in, and then put, there's a clear dental cement here, which is a bit hard to see, but there's dental cement around this. So it's glued to the skull with cyanoacrylic adhesive and then fixed in place with dental cement. So it's very simple, right? But on the other hand, it really helps because we can then record larger cohorts of mice relatively easily. And the idea that we're working towards is really to create a relatively open source framework for doing epilepsy studies in mice. And so there's some good recommendations about what we're supposed to do from an epilepsy standpoint in rodent models, but there's not really the equipment to do it. And so we've done a few things. We built an Arduino to control our experiments, and so it turns the lights on and off and starts and stops our experiments. We're designing a new preamplifying head stage, which will have an accelerometer rather than EMG wires in it. This is the first version. It just has 6 op amps on it, so we can record 6 channels. This is a commercial version that we're using at present, though, for our studies. And then there's a video of the mouse. The data's coming here. It goes to the analog to digital converter, to the computer, and you can see here there are 4 mice recording, and we have 4 video streams as well. So we have chronic video and chronic EEG, and this recording goes for 12 hours. It's restarted by this. It gets migrated to a primary server, and then a backup, and then a secondary server, which can do some data processing as well. And so in the case of epilepsy, it turns out to be really nice because this is looking at 12 hours of data across the screen, and this is not the best. I just made this slide this morning, so I was in a bit of a hurry, and I didn't know which mouse was the best, but oftentimes the mice have multiple spontaneous seizures in 12 hours. But the nice thing here is, I don't know whether you can see it very clearly, but there's a very broadband increase in power here, followed by a broadband decrease in power in the FFT, in the fast Fourier transform. This is just looking from 0 to 55 hertz here. And that's very characteristic of a seizure. So you see a massive increase in power with all these large amplitude spikes that are broadband because they're spikes. And then you see post-ictal electrographic suppression. So you see a decrease in power as well. And so you can identify these very easily. This is zooming in on the same thing. So you see a run of discharges, in this case in the hippocampus. So this is the EMG, the neck muscles. This is the left hippocampus. That's showing the same thing. This is the ECOG and the right hippocampus. And then we haven't done this. We've actually just had a paper accepted for sleep-wake. This is John Willie's lab, really, with Janos Exarchos, for sleep-wake analysis using machine learning, using a support vector machine and convolutional neural network. But we think that this will be even easier. We can make an SVM to detect seizures really easily, and then we can generate a report and do manual auditing and videographic analysis. Now, the really cool thing about this framework is that it turns out most people use videographic analysis for scoring seizures in rodents, and it turns out that it's easy to miss because seizures can be very subtle. So if there's a behavioral arrest in a mouse, you really don't know because they spend some of their waking time just sitting there anyway. But it turns out that the EEG is really, really sensitive. So even when there's a very subtle seizure, we see it electrophysiologically, and then we're going to compare that to our videographic analysis as well. Now that's all very nice. So we have a framework and an ecosystem for doing this sort of semi-automated long-term recordings of large cohorts of mice. We haven't yet implemented the seizure detection, but we don't think it'll be difficult. But that's not really addressing the main problem here. The problem here is that we need to do large-scale network recordings. And so we took a similar approach to that. So this is the same head plate now, but it has multiple microwire holes in it so that we can place microwires through the head plate. So we screw this in place, we can drop microwires. And the way that we can get them to the right depth is we made a capacitative touch detector so that when the microwire touches the brain surface, the light goes on. Then we can zero the stereotaxic frame and then lower it to the right depth in the brain. And this way we can target multiple nuclei. So here you can see there's like septal nuclei in hypothalamus as well as the hippocampi are being targeted in the entorhinal cortex, for example. And the way that this works is that you drill lots of holes with a tiny dental drill in the skull of the mouse. And then, I don't have a great photo set here, but this T-bar then sits on top of the head plate and holds the electrode interface board with an Omnetics connector on it. So this is the whole assembly in place. And this is, it's nice, you know, the head plate's glued in place with cyanoacrylic adhesive and dental cement around the bottom. And so it stays on nicely. And we've designed it for 32 microwires at the moment, but we can increase that as we use different head stages. So we're doing our next implantation of this on Friday, and I can let you know how it goes. We've only done one so far, so it's early days. But anyway, so these are the conclusions, really. So while local networks are really key for understanding epilepsy and other phenomena, we really need to look at large-scale networks, and we need to develop technologies to do that. And the problem with existing techniques is that they often, there are techniques for putting a lot of electrodes in the brain of a mouse, but they're not spatially segregated in a way that can record from a large-scale network. And they're really, therefore, neglected in at least mouse and rat science in epilepsy. And then if we look at a prototypical large-scale network of the sleep-wake network, it has multiple components that we could target to treat epilepsy. And then to really understand these large-scale networks, including in other things, so one of the other interesting questions, of course, is what's happening at a network level when we make a transition from sleep to wake? What's driving that? So I'm interested in studying those questions with this technique as well. So this wouldn't have been possible without a large number of people who have been very helpful and great colleagues and collaborators. And foremost people in the lab, so I mentioned Adam Dickey. Lauren is our lab manager. These are undergraduates in the lab who have really been outstanding, and Austin's a programmer who's actually moved on. Now, it's really hard to retain a programmer in a lab, isn't it? They get paid too much in industry. We also have a large number of collaborators as well. So Dan Drain is someone I speak to every day, and David Gutman is working on us with a DTI project, actually looking at CSEPs and DTI correlations. These are our neurosurgeons who implant the patients and also collaborate on a basic science level. David Weinshanker has been a great collaborator in relation to sleep-wake studies, as well as Andrew Eskai for epilepsy, and Heidi, Sean, and Bill has made the new preamplifiers for us, so he's got an electrical engineering background. I still keep in close contact with people, Beth Israel, and then I have a variety of mentors as well that have been very helpful in my development at the moment as a junior faculty member. So thanks for your attention, I appreciate it. Yeah. Are there
time for one question?
Richard?
So you mentioned very briefly about patients when they are undergoing a seizure who may be amnesia. Is this a general finding that they tend to remember the seizure itself even though they may be screaming their
So yeah, so it's good to think about the networks of memory. And so if their memory networks are involved, then they don't remember it. So oftentimes patients with medial temporal lobe seizures don't remember their seizures. Sometimes patients with unusual frontal lobe seizures will be enacting a complex series of behaviors, but will remember what's going on and will be aware. So it really depends. And generally, related to the amnesia is a loss of awareness as well, and these are two separate phenomena. But there are kind of electrophysiological and circuit bases for those. So it depends on the seizure type. Yeah.
Thank Nigel again for this.
Thank you. Thanks very much.